The main idea, well, I start from the beginning. Why we start this project? It's because we wanted to create a DNA-sensitive biosensor which could detect the DNA sequences of infected disease. And we start our studies from sequences from pH-positive leukemia type, which is the product of connection of two genes. Its mutation causes the formation of leukemia in human organisms. So we started such work. There is a theory that when quantum dot is close enough to gold nanoparticle, there became a process of energy transfer from donor to acceptor, where quantum dot is a donor and gold nanoparticle is an acceptor. And when they located 10 or 9-10 nanometers close to each other, should start the process of energy transfer, and it could be visible of optical characteristics of quantum dot. They like fluorescence is very like have intensive, but if it's with gold nanoparticle in solution in such distance as 10 nanometers, it became lower, and the dynamic of reaction should be like lowering during like some time of incubation period. This like was the main idea, that we put two types of nanoparticles in solutions, modified them with oligonucleotide, which are complementary to the leukemia gene, and the long length of oligonucleotide should be such to help locate two types of nanoparticles close enough to make this energy transfer process real. But when we start our experiment, we have such problems, which is electrolytic aggregation of gold nanoparticles. It is known fact that gold are very sensitive for saline solutions, such as like different types of buffer, etc. But the reaction needs a buffer solution. So, we stabilized our gold nanoparticles with tiols, such as mercaptahexanol, and with lipoic acid, which is a sulfurous molecule, which is sulfur very good connected to gold always. These tiols and lipoic acid and also chelated oligonucleotide make gold more stable in solution, so it could be longer water soluble, and there shouldn't be any electrolytic aggregation. But still we have such problem, and we start to study it, and it was very weird results that we had, because like in normal condition, if there is no quantum dots in solution, gold nanoparticles, they wouldn't aggregate in buffer solution. They like, okay, they are water soluble and everything is all right. But if we add quantum dots to solution, which are modified with short oligonucleotide, we have very strong like indexes of electrolytic aggregation, which shouldn't be. And we have like some interaction between these nanoparticles, which we still don't know why it like appears, because it shouldn't be. We have such energy transfer process, because we can see that the photoluminescence of quantum dots is quenching when we add gold nanoparticles modified with the oligonucleotide, which is like not supposed to be. So it's like this work like says that this theory is not perfect because they shouldn't like interact each with other if there is no short distance between them. But we have like they're free in solution. They're only both of them modified with the oligonucleotides, and they somehow find the way to interact with each other without any leukemia sequences. So it's only this short oligonucleotides. Like we supposed to suggest that there will be like quantum dot with short oligonucleotide, gold nanoparticle with short oligonucleotide, and this short oligonucleotides, they should be complementary to a free soluble sequence of leukemia gene, and so then they hybridize, and we have quenching of photoluminescence. It was the perfect theory, which is not working. So that's, well, I think that's it. What I wanted to say. If you have any questions.
Of course, thank you so much, Evgenia. Yes, we have one question in our chat from our co-chairman. It's from Larissa Skivka. So does this nanosensor specific for the only one nodological form of leukemia or not?
Well, actually, yes, but it was like a tested project. We want to find out if it will work out, like well, if it will succeed. So we want to check this theory because it's like very popular to work with quantum dots in our times. So we want to like check this theory if it's real or not, and we start from leukemia because it's a comfort model to work with.
Thank you so much. So we just don't have unfortunately time for more one question.